Assalamualaikum and morning everyone. So today we are going to continue our topic, which is regarding the neurophysiology, which is the neuron, okay, neuron structure and function. Okay. Before that, let me share my slide first. Okay, I hope all of you can see my slide. Okay, so today we are going to continue about this topic of diversity of neural signal. Okay, neural signaling. So in the previous lecture, we already learned about the electrical signal in neurons, which is we have the membrane potentials. We also already learned about the graded potentials, the action potentials. Okay. We also already learned about the voltage-gated channels, which is involved during the action potentials, which is the sodium channels and also potassium ion channels. And you also learn about the transmission signals across the synapse, which is you have the synaptic cells, post synaptic cells, and also synaptic cleft. Okay, and if you still remember, also we already learned about the transmissions at the neuromuscular junctions that release the acetylcholine. Okay, so there is the part of subtopic that we already learned in the previous lecture. So now we move to the another subtopic. Okay, still in the neurophysiology topic. Okay, the neuron function and structure. So this is the variation in synaptic transmissions. Okay, so if you recall back that the AP in the neuromuscular axon terminates by triggering the release of neurotransmitter. Okay, that we call as the acetylcholine. Okay, ACH, which ultimately triggers the muscle contraction. Okay, so this is the steps, the process, okay, where it starts with the acetyl CoA synthesized in the mitochondria. All right. So it goes to the process of it will transfer the catalyzed conversions of choline and acetyl to the ACH, okay, from acetyl plus choline to the ACH, okay, and inform the synaptic vesicles, okay, so the ACH are released into the synapse, okay, which is the acetylcholines bind to its receptor, okay, and they have the acetylcholinesterase, okay, which is breaking down the acetyl into choline and acetate, okay, and the pre-synaptic cells, which is take up back, okay, the recycle of the choline. Okay, so this is the process of the process that involved in the neuromuscular axon, okay, which is release the neurotransmitter, which is acetylcholine. Okay, so this will trigger the muscle contraction. Okay. However, the synaptic transmissions is incredibly diverse. Okay, not only specific to that particular process. Okay, so the animals also use different types of transmissions to accomplish the different types. Okay, so we have different types of synaptic transmissions in order to accomplish the different tasks. So some neurons, they don't even use the neurotransmitter, okay, in signaling, but they are instead directly connected to their target cells, okay, via the gap junctions. Okay, and the synapse where the presynaptic cells and the postsynaptic cells are connected by the gap junctions, okay, we call as the electrical synapse. Okay, but to difference with the chemical synapse, okay, which is they use the neurotransmitter in signaling. Okay, so this is the gap junctions. Okay, whereas it transmit the signal through the gap junction. So this is the variation in synaptic transmissions. Okay, we have two signals. We have the electrical synapse. We have the chemical synapse. So the electrical synapse is the signals are sent directly from the presynaptic cells to the postsynaptic cells through gap junctions. Okay, so this is the electrical signals. While for the chemical synapse, okay, the chemical synapse, the signal is transmitted via chemical neurotransmitter. Okay, which is we have the for example, we have the acetylcholine. Okay, in the transmission in the neuromuscular junction. Okay, for example, so which across the synaptic cleft. So this is the synaptic cleft and bind to the receptor on the postsynaptic cells. Okay, to induce a response. So this we call as the chemical synapse. So the synaptic transmissions that using the gap junctions, okay, they produce they have the electrical signal. Okay, while the signal that transferred, okay, that transmit via the chemical neurotransmitter across the synaptic cleft, okay, we call as the chemical signal. Okay, so this is the variation in synaptic transmission. So both of the synapse, which is the electrical and chemical synapse, they have a difference in a numbers of way. Okay, which is the first one, we have the different in directions of flow of the informations, and the second one we have the speed of variation in the speed of transmission. Okay, so we go to the first differentiation, which is the directions of flow of the informations. So in the chemical synapse, okay, in the chemical synapses, the information flow in a single direction. Okay, satu direction only. That is from the presynaptic. Okay, this is from the presynaptic. So this is the presynaptic cell to the postsynaptic cells. Okay, only one direction. Okay, while in the electrical synapse, okay, the informations can flow in both directions. Okay, from the presynaptic cells to the postsynaptic cells via the gap junctions, or from the postsynaptic cells to the presynaptic cells through the gap junctions. Okay, because of the pre and postsynaptic cells, they are directly connected. Okay, directly connected by the gap junctions. Okay, it difference with the chemicals synapse, which is they only have one way of direction. Okay, one way of flow of informations, such which is from the pre to the post. All right. So the electrical synapse and chemical synapse also they are differ in number of speed of the transmission. Okay, just now you're talking about the differentiation in the directions of flow of the informations. Okay, and the second one, they are also different in the speed of transmission. So in the electrical synapse, they are fast. The transmission of the signal are fast, very fast. Okay, while in the chemical synapse, they are very slow. Okay, so they are slower than the electrical synapse. Okay, because of the delays associated with the diffusions of the neurotransmitter. Okay, across the synaptic cleft, and also they are associated with their binding to the receptor on the postsynaptic neuron. So this is the process that delay the transmission of the signal. Okay, so basically the speed of the transmission, the electrical synapse, the speed of fast, okay, faster compared to the chemical synapse. So the major benefits to use the chemical signal is the diversity of signal that can be conducted. So even though the chemical synapse are slower compared to the electrical synapse, but they also have the benefit, okay, which is the diversity of signal that can be conducted. Okay, there is a lot of variation in both structure of the chemical synapse and in the types of neurotransmitter. Okay, so even though they are slower, but they have benefit. All right. So this is based on the types of synapse, which is for example in the axon terminal. Okay, in the axon terminal or the axon varicosities. Okay, and in the unfastened synapse. Okay, you see unfastened synapse is actually where one where one neuron joins at the place beside the axon terminal. Okay, sebelah beside the axon terminals. Okay, so they have the same functions and effect, merely in a different place only. Okay, they are side by side. Okay, so this is the unfastened synapse and also in the spine synapse. So all of these are referred to the different synapse structure and therefore different function in cells. Okay. So each of these types of synapse can also be found in different locations around the neuron. All right. So for example, this is the location of synapse. For example, we have the dendritic synapse. Uh, we also have the exoaxonic synapse. Okay, which is the transmission of the signal happen, okay, in the axon in hop. We also have the exosomatic synapse. So exosomatic synapse is where the transmission of the signal, okay. The location of synapse is happen in the soma, okay, body of the cells, and also in the exodendritic synapse. So exodendritic synapse is where the transmissions of the signals are happen in the dendrites. Okay, so all of these are referred to the different synapse locations. So the neurons are unique to animals. Okay, so neurons are only found in animals. Okay, the neurons can only found can only found in animals. Sorry. Yeah, though the plants, they do have the sensory receptor. Okay, they also have the ion channels, action potentials that can process the informations, but they don't have the neurons. For example, in the Venus flytrap, okay, so the Venus flytrap catch their prey when the sensory hair are stimulated. Okay, so this will trigger the AP, the action potential, which spread through the cells via the plasmodesmata. Okay, which the informations are spread out through the plasmodesmata. So this plasmodesmata is actually in the similar structure to the gap junctions. Okay, which is it send the electrical impulse. Okay, the electrical signals. Okay, the plasmodesmata are very similar structure to the gap junctions. Okay, so this will cause the trap to shut. Okay, the trap to shut. So just now we are talking about the diversity in signal. Okay, diversity in signaling. So now we are moved to the second subtopic, which is the diversity in neurophysiology, the physiology of the neuron. So although all the neurons have the same basics component, okay, so we have four basic components, okay, which is if you remember, we have the R, I, C, T. Okay, we have the reception phase. We also have the integration phase. We also have the conduction phase and transmission phase. Okay, although all the neurons have the same basic components, which is these four components, each of these components has been modified by evolutions in order for them to have the better performance to this specific task. Okay, so all the neurons have the dendrite. They also have the cell body, the soma, and also they have the axon. Okay, but details of each structure are variable. Okay, so this is the examples of neuron diversity. They have a different structure of the neuron, the diversity of the neuron. Okay, the neuron physiology. So this is the structural diversity of the neuron. Okay, so the details of each structure are available. So, okay, for example, the Purkin cells. This is the structure of the Purkin cells of mammalian cerebellum. Okay, they have a lot of dendrites. This is the vertebrate motor neuron. Okay, so this is the neuron from the cerebellum. Okay, so this is the neuron in the vertebrate retina. So this is the neuron, the structure of neuron in the vertebrate olfactory neuron. Okay, so this is the insect mechanoreceptor neuron looks like. So this is the insect sensory neuron looks like. So this is the structure diversity of neuron. They have a diversity. They have a different structure of neuron. So the neuron can be classified based on function. The neuron also can be classified based on structure. Okay, so we go to the function first. Okay, so this is the functional clusters of neuron. We have the efference neuron, interneuron, and sensory neuron. Okay, so the motor neurons, which is the efference neurons and lower motor neurons, they are multipolar. Okay, multipolar shape cells. In order for them to conduct action potential out of the central nervous system. So this is the functions of motor neuron, the effector neuron. Okay, so the interneurons, these interneurons are located within the central nervous system, the CNS. So the functions of the interneuron is to convey the signal from one neuron to another. All right, they are functioning in conveying the signal from neuron to another neuron. And the neuron that based on the function, which is the sensory neuron, the efferent neurons, okay, they are unipolar, bipolar, or multipolar shape cell, okay, the sensory neurons that conduct the action potential towards or into the central nervous system. Okay, so this is the sensory neuron. Okay, so the structure can be unipolar or bipolar or multipolar. Okay, in order for them to conduct the AP towards or into the CNS. So this is the classes of neuron that based on the function. Okay. We also have the neuron that can be classified based on structure. Okay, the structural classes of neuron. We have the unipolar, bipolar, and multipolar, and some also we have the pseudo unipolar. Okay, so the unipolar neurons have only one structure that extend away from the soma. Okay, they have the structure that extend away from the soma. Okay, so these neurons are not found in vertebrates but are found in insects where they stimulate the muscles or gland. Okay, and some of the unipolar neurons are also sometimes they call as a pseudo unipolar. Okay, because they originate the embryologically as the bipolar neurons. Okay, some they are called as a pseudo unipolar. So most sensory neurons are pseudo unipolar. Okay, and have an axon that branch into two extensions. Okay, one connected to the dendrite that receive the sensory informations, and the other one is transmit this information to the spinal cord. Okay, one receive, one transmit. Okay, and the second classes of neuron that based on the structure, which is the bipolar. So the bipolar neuron has one axon and one dendrite extending from the soma. And examples of a bipolar neuron is a retina bipolar cells. Okay, so this is our examples of the bipolar. You also have multipolar. So multipolar neurons are the most common types of neuron, which is each multipolar neuron contains one axon and multiple dendrites. Okay, they have one axon and multiple dendrites. Okay, and multipolar neurons can be found in the central nervous systems. Okay, and the CNS, which is in the brain and spinal cord. Okay. So this is the classifications of neuron that based on this structure. Okay, and again we have unipolar, bipolar, and multipolar, and we also have some sort of pseudo unipolar. Okay, so can you classify the structure of this neuron that based on the functions? Okay, so which neuron are located in the brain. Okay, so they are this one and this one. Okay, so this is the structure of neuron. Okay, which is located in the brain. Okay, because they have a lot of dendrites, and these are the neuron that are located in the sensory. All right, and this one is the structure of neuron, right? Okay, they're located in the muscle. Okay, whereas they have the myelin sheath. Okay, myelin sheath. Okay, in order to allow the signals that travels to the long distance. Okay. Okay, so we go to the first diversity of neuron. Okay, neuron physiology, which is the sensory neuron. So the sensory neurons are found in animal sense. Okay, for example, insight, hearing, touch, taste, smell. Okay, and at one end of the neuron is a receptor. Okay, so they have at one end they have a receptor. Okay, that is associated with the with that particular sense. Okay, either that associated with the sight sense or touch sense, taste sense, smell test, smell sense. Okay, for example, the olfactory receptor. Okay, for example, is the olfactory receptor. Olfactory is regarding the smell. Okay, so it involved in the smell are activated by airborne chemicals. Okay, so at the other ends of the neuron, okay, they have a lot of dendrites, okay, that allow the sensory neuron to connect to the brain for processing, to transmit the signal to the brain. Okay, so this is the sensory neuron. We also have the brain neuron. Okay, so the brain neuron is type of neuron that called the interneuron. So they have a large number of dendrites, okay, on both ends, okay, to maximize the connection with other neuron. Okay, so you can see from here they have a large number of dendrites. Okay, then they are often lack of obvious axon. Okay, so dia tak ada axon nampak panjang tu because they are only transmitting the signal over short distances. Okay, between other neurons densely packed in the brain. Okay, so all of these neuron they only can be found in the brain. Okay, and also they are lack of axon. Okay, so the other types of neuron, which is the vertebrate motor neuron. So they have a long axon cover in myelin sheath. Okay, so this is the axon. Okay, the axons are covered by the myelin sheath, the purple or blue color. This one. So this will allow the signal to travel along a long distance. Okay, so at one end of the branches, they are branched into the neuromuscular junctions, and the other end they have a lot of dendrites. Okay, which is for connecting the muscles to the brain or to the spinal cord. All right. So the diversity in neuron signaling can also be achieved by varying cells that associated with each neuron. Okay, so this we call as the accessory cells or glial cells. So for example, in the vertebrates, there are 5 types of glial cells, the accessory cells. Okay, which is the first one we have the Schwann cells. The second one we have the oligodendrocyte. The third one we have the astrocyte. The fourth one we have the microglia, and the fifth one we have the ependymal cells. So all of these are the glial cells or the accessory cells, okay, in the neural signaling. Okay, so we go one by one. The Schwann cells, which is actually a form of myelin sheath, okay, and these are associated with the neuron with long axon. Okay, so this is the Schwann cells. Okay, the blue color it is actually a form of myelin sheath. Okay, so the function is to increase the conduction speed and prevent the decay of the action potentials. Okay, so this is the function of the Schwann cell. So we go to the second accessory cells, which is oligodendrocyte. So this is the oligodendrocytes. Okay, so this is the oligodendrocytes. Yeah, it actually form myelin sheath in the central of the of the central nervous system. Okay, so dia pun actually a form of myelin sheath, but they are located in the central nervous system. Okay, so we also have the third accessory cells, which is astrocyte. So this astrocyte, so this is the astrocyte, the green color. Okay, so the astrocyte is found in the central nervous system, still in the CNS. Okay, so this is the BNS, so this is the CNS. So the astrocyte is found in the CNS and have a numbers of functions, which is including the transport of nutrients and the neuron development. Okay, so this is the function of astrocyte. The fourth accessory cells, okay, which is microglia. So the microglia is here, the purple color. Okay, so the microglia are the smallest glial cells, and they are involved in neuron maintenance, which is functioning to remove the debris and dead cells. Okay, so this is the function of the microglial cells, the smallest glial cells, the smallest accessory cells. Okay, and the last one we have the ependymal cells. So these ependymal cells here, they can be found in fluid-filled cavities of the CNS. All right, and they are often ciliated, which is they have the tiny hairs and circulate the spinal fluid. Okay, the function is to circulate the spinal fluid. So this is the five accessory cells or the five glial cells in the vertebrates. Okay. So we go to the Schwann cells. Okay, the Schwann cells is a form of myelin sheath and are associated with neuron with a long axon. So this is the axon. All right. So these accessory cells, okay, the glial cells, uh, the myelin increase the conduction speed and prevent the decay of action potentials. Okay, so this is the function of Schwann cells. Okay, so as you can see from here, the Schwann cells wrap around. So this is the Schwann cells. So it will wrap around an axon. Okay, so the chocolate color is the axon. So the Schwann cells are wrapped around the axon many times. Okay, in order to form the myelin sheath. That is why the Schwann cells is actually form of myelin sheath. Okay, so the function is to increase the conduction speed and to prevent the decay of the AP. Okay, so the invertebrates lack of a true myelin sheath, but they are instead are wrapped in membranes of glial cells called gliocytes. Okay, even though they don't have the Schwann cells, which or the form of myelin sheath, but they have the gliocytes. Okay, in the invertebrates. So this is the evolution of myelin sheath. Okay, so certain invertebrate neurons are wrapped in multiple layers of cells membrane similar in appearance to vertebrate myelin sheath. Okay, even though they are lack of myelin sheath, but they have the, they have the wrap in multiple layers. Okay, of the membrane of the cell membrane. Okay, they are similar with the myelin sheath. Okay, so this include the nerve fiber in the ventral nerve cord of shrimp. Okay, crabs or earthworm. Okay, so the proteins of the protein complex, okay, we call at the septate junction, okay, in the invertebrates. Okay, so the certain junctions hold cells in place as they wrap around the invertebrate axon. Okay, so in the vertebrate, the thing that the cells that wrap around the axon is the Schwann cells, or if there are multiple. Okay, if they are wrapped many times, they will form a myelin shelf in vertebrate. Okay, but in invertebrate, they are called septate junctions. Okay, so these septate junctions structure suggests that invertebrate wrapping play a similar role to the myelin shelf in the vertebrates. Okay, but even though they have the similar role, but they are evolved independently. So this is the evolution of myelin sheath. Myelin sheath. So still in the evolution of myelin sheath. Okay, the invertebrate versus the vertebrate axon wrapping. Okay. As you know, in the vertebrate there are form of myelin sheath. Okay, in the invertebrate there are some sort of septate junction. Okay, so the layer of membrane in the invertebrate wrapping are not as closely stacked as the vertebrate layers of myelin sheath. Okay, so they are not as stacked. Okay, tak rapat-rapat macam gitu. Okay, so the protein compositions of myelin sheath is different. Okay, so the protein are critically important forming myelin sheath are largely missing from the invertebrate axon wrapping. Alright, so in the vertebrate they have a protein. Okay, and a wrapping likely evolved separate by the convergence evolutions. So the convergent evolution is where the process, okay, the process whereby the organisms that is not related, that is not closely related, there is not there is not monophyletic, okay, independently evolve. Okay, they are evolved as you can see from this example. Okay, so this is the animals that they are not related, but they are independently evolved. They are similar trait. Okay, for example, in here the trait of flying. Okay, they are independently evolved the similar trait as a result of having to adapt to similar environment. Okay, so for example, in here the wing of the birds, the reptiles, okay, and also mammals. Okay, all are used for flying. Okay, the traits for fly, but they are evolved independently and thus they have different structure. Alright. So we go to the myelinated axon. Okay, so the axon, the myelin sheath, the myelin sheath. Okay, so this is we call as the apa tu we call as the myelinated axon. Okay, myelinated axon. So the true myelin sheath were an important evolutionary innovation. Okay, and they are allowed rapid signal conduction in a small amount of space. Okay, the true myelin sheath. Okay, because they are wrapped around the axon. They are this is axon. Okay, they are wrapped around the axon many times. Okay, this is why they can allow the rapid signal conduction in a small amount of space. Okay, and also the true myelin sheath also important, okay, to help to provide the conditions for complex vertebrate nervous system. Okay, so more complex nervous system, okay, if the animal they have more complex nervous systems, they will allow the animal to evolve more complex behavior. Okay, they also can allow the animals to evolve more complex of physiology, social systems. Okay, that is why the animals need to have complex nervous systems. Okay, for example, in here, from the lampreys or hagfish, they don't have the myelin sheath. Okay, but as they are evolved, okay, the mammals they have a lot of myelin sheath. Okay, so the mammal they have a very complex nervous systems. Okay, so when they have the very complex nervous system, they will have the more evolutions of behavior, physiology, social systems, and etc. Okay, so still in the myelinated axon. So the nervous system that rely on large diameter axon, okay, the diameter axon. So this is the axon. Okay, they are rely on the diameter of axon. Okay, so the diameter of axon, the diameter started from here. Sorry, the diameter of axon, and less effective axon wrapping are simple relative to complex nervous systems in invertebrate. Okay, so the invertebrates have simple nervous system that typically lack a central processing like a brain. Okay, for example, this is all the invertebrate. So they have a lack of CNS. Like in the molars, they have a small brain, but in the cnidaria, the flatworm, they don't have a brain. They are lack of brain. Okay, but even though they have a very simple nervous system, but they are actually functioning like the complex nervous system which we seen in the vertebrates. So we know that the neuron are differ in the speed, okay, at which signal are transmitted, right? So the axon conduct action potential at different speed. Okay, in the axon, the conduction of the AP they have a different speed. Some are quick, some are slow. Okay, so the speed of the action potential, okay, difference by two variables, which is the presence of the myelin. Okay, because of we know that the true myelin, they can allow the rapid signal conduction, okay, in a small amount of space. Okay. So the second factor, okay, the second variables is the axon diameter. All right, either the present or not of the myelin or the axon diameter. So this is can influence the speed of the AP. Okay, for example, in here, so this is the conduction velocities in axon from various species. For example, you have this squid. Okay, so they have a giant axon. Okay, the axon sangat besar. So the diameter is 52,000 of micromillimeter. Alright, so they have a big or giant axon, but they don't have the myelination. And the speed, okay, the speed of the action potentials, the speed of the propagations, which is 30 meter per second. All right, like second example in the crayfish, okay, they have the 36 diameter of the axon, and they don't have the myelin, so the speed will lower compared to the animals that have bigger axon diameter. Okay. So we look through the this one, the frog. Okay, the frog, which is in the A nerve, A in the sciatic nerve, which is in the A fiber. So they have a very small of diameter of the axon, but they do have myelin, right? And you see, the speed are higher compared to the axon, compared to animals that have the giant axon. Okay, so the speed of the AP is actually depends on either the presence or not of the myelin, and it depends on the axon diameter. Okay, if like this one cat, okay, in the cephalus nerve C fibers, so if the cat they only have small diameter of the axon and they don't have the myelin sheath, so you see the speed of the propagations are very low, which is only 2 meters per second. All right. So, still in the diversity of neurophysiology, we have the Ohm's law. So, this Ohm law describes the speed of the AP traveling down the axon. Okay, the speed of the AP are traveling down the axon. So, the speed we call as the current, okay, I. So, this is the speed of the AP that traveling down an axon. So the speed or the current of the signal depends on two variables, which is voltage or resistance. Okay, so the current, the speeds are depends on the voltage and the resistance. Okay, so essentially the strength of the signal along the axon, okay, which is the current, is greatest. Okay, the current is greatest when the voltage, okay, which is the input of the energy, is high and the resistance is low. So this one will provide the high of the signal of the X1. Okay, signal of the highest current. Okay. So the resistance, okay, the resistance, which is the force opposing the conduction, is applied by different components of the cell. Okay, so this is the different component of the cells, the extracellular fluid, the membrane, and the cytoplasm. So each component it has a different resistance value, which will reduce the strength of the signal over the space. Okay, starting from the strengths are going down from the extracellular to the membrane to the cytoplasm. So each compartment here they will reduce the strength of signal. Okay. So cable properties. Okay. So the similar physical principles, okay, like from like by the govern current flow through the axon and the telephone cables. So the govern current flow through axon and the telephone cables, they have the similar physical principles. Okay, so the current, all right. So the current is the amount of charge moving past a point at a given time. Okay. So a function of the drop in voltage, okay, so voltage is actually the input of energy. So a function of the drop in voltage across the circuit and the resistance of the circuit. Okay, it depends on these two factors. Alright. So the voltage is energy carried by a unit of charge, and the resistance is the force opposing the flow of the electrical currents. So the Ohm law is the voltage equal to IR. Okay. So still in the cable properties. Okay. So the ion moving through the voltage gated channels cause a current across the membrane. Okay. So the current across the membrane. Okay. The current across the membrane. Okay. So the current spread electronically. Okay. And some currents leaks out of the axon and flows backward along the outside of the axon and to completing the circuit. Okay. So this is the current flow in the axon. All right. So this is the extracellular, the membrane, and cytoplasms. So still this is the extracellular, the membrane, and the cytoplasms. Okay. So this is the electrical circuit model for a patch of membrane. All right. So each area of axon consists of an electrical circuit. So they have the three resistors. Okay. So they have the first one is the extracellular fluid. Okay. The first resistor is the extracellular fluid. The second one is the membrane. Okay. The membrane and the cytoplasms. Okay. The cytoplasm resistor. Okay. So this is the electrical circuit model for a patch of membrane. Okay, so we go to the myelin sheath. So again, the myelin sheath is prevent the signal from traveling through this area of high resistance. Okay, so as a result, the current or conduction speed will increase. Okay, so this is the function of myelin sheath. Okay, so this is the myelinated axon. Okay, so the speed of conduction and resistance. So the axonal conduction is combination of the electronic current flow and APs. Okay, so the electronic current flow is much faster than the APs, right? Okay, but the electronic current flow is graded and can travel only short distance. Okay, compared to the AP, it can travel along a long distance. Okay, so the greater lambda, so the lambda is actually the length constant of the membrane. So the greater of the length constant of the membrane, more electrical current will flow. Okay, so just now is the speed of conductions and resistance. So this is the speed of conduction and capacitance. Okay, so biological membranes are act as electrical capacitors. Okay, so the capacitance is actually the quantity of charge that needed to create a potential difference between two surfaces of capacitor. Okay, so this depends on three features of capacitor, which is the material properties of the capacitor, which is generally the same in cells, or the area of the two conducting surface, which is the higher area. Okay, they can have the higher capacitance, or it depends on the thickness of the insulating layers. Okay, more thickness of the layer, insulating layer, they have the lower capacitance. Okay. So still in the speed of conduction and capacitance. So the time constant T is actually the time needed to charge the capacitor. Okay, so the T is actually equal to the membrane resistance and membrane capacitance. Okay, so the increase in either membrane resistance or membrane capacitance will increase the time constant of the membrane, and it will delay the current flow across the membrane. Okay. So the giant axon. Okay, so the giant axon, they have the high conduction speed. Okay, because they have the larger diameter. Right. So this giant axon easily visible to the naked eye and can be built up to millimeter in diameter. Okay, so this giant axon not present in mammals. All right. For example, in here, how does it works? So the mantle cavity fills with water. So the mantle are filled with water. Then the brain in here, they send a signal to the satellite. Okay, so the stellate ganglia, which send signal along the axon. Okay, so this is the stellate ganglion. Okay, so it will send the signal along the axon. Okay, along the axon of the different diameters in the mantle. Okay, so the nerve impulse reach the muscles at many points in the mantle cavity. Okay, so then the muscles of the mantle will contract synchronously bersama-sama, sama-sama serentak, and rapidly closing the mantle, so it will forcing the water out from the siphon. Alright, and this one can produce rapid jet of proportions. Okay, so this is how the diameter of the axons, okay, give the influence to the animals. Okay, so the same can be said for the large diameter axon, which is less of the axon surface area is exposed to the membrane and cytoplasms. So if the animals have the giant axon, so the less of axon surface is exposed to the membrane and cytoplasms that slow down the signal. So the resistance is low. Okay, so the animals that have the giant axon, they will have the lower resistance. So as a result, the current or the conduction speed will increase. All right. So the consequences of increasing the ion axon diameter. So although the increasing of axon diameter provide the faster conduction velocity, they are also have two main disadvantage, okay, in using the large axon to increase the conduction velocity. The first one is the large axon take up more space. Okay, so this is the first disadvantage of having the large axon, and this may limit the number of neurons that can be packed into the nervous system, the CNS. Okay, for example, mammals have nervous system densely packed with neurons and accessory cells. So the mammals cannot afford to lose additional space. That is why the axons are not happen in the are not present in the mammals. Okay, the giant axon, they are not present in the mammals. And and then the second disadvantage of having the having the bigger axon, which is the large diameter axon, have a much larger volume of cytoplasm per unit length. Then it will make them to be energetically expensive to make and maintain. Alright. So the cytoplasm, because of the cytoplasm, they contain all of the organelles such as the mitochondria, and many energy depending processes occur in the cytoplasms. So the bigger cytoplasms, they will have the energetically expensive, okay, because they need to more energy to make and maintain. All right. So that is the two disadvantage of having a big axon, okay, giant axon. And as a result, the giant axons are typically used when extremely high speed conduction is necessary for survival. Okay, they are not use the giant axon, every time. Okay, they are only used when they are extremely need for the survival. So, for example, in squid, the axon control the rapid contractions of the mantle used during escape and prey capture behavior. Okay, for survival only, they use the axon to control the rapid contractions. Okay, so not all muscles, okay, in the mantle will contract during the basic swimming. Okay, only when they are need for survival, either to escape from the predator or for the prey capture. Right, then we have already end of this topic, okay, which is the neurophysiology, the neuron structure and function. So let we summarize our class for today. Okay, the first one, once the AP reach the axon terminal, okay, they have the axon terminal, so the neuron must transmit the signal across the synapse, okay, to the target cells. Okay, and in muscles, they involve of the flow of calciums and neurotransmitter, the chemical of neurotransmitter, which is acetylcholine, okay, in muscles. So all neurons, they have dendrites, the structure of neurons, they have dendrites, they have the cell body, which is soma, and also they have the axon. But details of its structure are variables. Okay, for example, the interneurons, okay, the neurons that have a lot of dendrites, okay, so the interneurons in the brain, they have very short axon and many dendrites. right? In the sensory neurons, they have a receptor on one end and dendrites on the other. Okay, they have a receptor. Okay, at one end they have and also they have the dendrites. Okay, at the other end. While the motor neuron, they have a neuromuscular junctions. Okay, even though they have the same structure, but each details of structure are variables. Okay, then the Schwann cells, okay, the accessory of the cells or the glial cells, it's actually form of myelin sheath. Okay, so this functions to prevent the decay of the AP when traveling down the axon. We also have the Ohm's law. Okay, so the Ohm's law are describe the speed, okay, the speed of an action potential traveling down an axon. So the signal travel faster down myelinated and large diameter axon because the resistance is low. Okay, so the speed can be more fast if the resistance is low. So then we have the increase in axon diameter and axon myelination increase the speed of conduction. Okay, the animal that have a large diameter of axon and they have the myelin, so they have the biggest speed of conduction. And the squid, they use the giant axon to ensure the mental tissue contract simultaneously. Okay, and the large axon take up more space and it require more energy to maintain. So this is the two advantages of having the large axon, which is they take up more space and they require more energy to maintain. And the vertebrate are used the myelinations to increase the conduction speed and innovations that help to develop the complex CNS. Okay, and the neuron signal are also varied by the differences in the synaptic transmissions. Okay, and the animals can alter the types of synaptic transmissions, either the electricals or chemicals. And also they can alter the structure of synapse, the location of the synapse, and the type of the neurotransmitters. Okay, it depends on the functions. And then last, this diversity in neurosignaling is necessary for the complex nervous system found in the vertebrate. Okay, so that's all for today's lecture. So, and again, you need to answer the questions, okay, the exercise. Okay, so you need to combine with the Monday class, all right? So for today, you only have 4 questions to be answered. The first one is the ABC. Which of the following statement is false? Okay, either A, B, C, or D. And the second question is you need to list and describe the functions of the structural components of the neuron. Okay, and the third one, you need to list and describe the 4 3 plus 1. Okay, the 4 3 plus 1 types of neuron, which is we have the unipolar, bipolar, multipolar, plus the what, pseudo polar. Okay, you need to list and describe. And the last questions, you need to compare the functions of different types of glial cells, which is we have 5. For example, we have the swan shell, the aristocyte. Okay, so you need to compare compare the functions of different types of glial cells, the accessory cells. Alright, so that's all from me for this class. Please do the exercise, which is only 4 questions for today's class, then combine with the Monday class. Okay, then you need to submit. Alright, that's all for me. Assalamualaikum and see you next week.